Okay, so now we're going to talk about meiosis. And meiosis is a different type of cell reproduction because meiosis produces the gametes. So it's going to produce the sperm and the egg cells. Meiosis is going to produce 4 daughter cells instead of 2. Sperm cells are called daughter cells too, so if it's producing sperm, they are called daughter cells. These daughter cells are going to be genetically different from the parent cell and from each other. So that's a really important part of meiosis. That is really why we see so much variation in plants and animals that sexually reproduce, because these daughter cells are each different. So a cell that undergoes meiosis is going to start out as a regular, what we call diploid cell, and so it has a full set of chromosomes. It's going to go through the cell cycle and have a G1, an S, and a G2 phase, but that in that replication phase, the chromosomes are going to they're going to be replicated, and they're going to have sister chromatids. But I wanted to just point out to you that there's homologous chromosomes within the cell. Homologous chromosomes are going to be the chromosomes that have the same genetic material, the same genetic information, but the mother's information is one of the chromosomes and the father's information is the other chromosome. And so we call those homologous chromosomes. I'm going to walk you through this graphic that shows the phases of meiosis. Meiosis, a lot of the same things happen that happened in mitosis, but in meiosis there are 8 stages instead of 4. And so on this graphic, they show here the interphase. This is not actually part of meiosis. Interphase is part of the cell cycle. It's shown on here to show you that cells that go through meiosis, they still go through the cell cycle. But meiosis actually starts at prophase 1. And in prophase 1, a lot of the same things happen here that happen in mitosis. So the nuclear envelope is breaking down, the chromosomes have condensed. One of the things that's different, though, is that these homologous chromosomes are joining up together. And so there's a matching chromosomes from your mother and father that have paired up. So instead of seeing just one chromosome set here, one duplicated chromosome set, you're going to see homologous chromosomes. And so there's going to be this set like this that you will start to see in prophase 1. And something really important happens here. It's called crossing over. So I'm going to go to this graphic. You have the homologous chromosome pair, and then as the chromosomes get closer together, what we see happen is a synapsis forms. And that's right here where these non-sister chromatids, these are called non-sister chromatids because these 2 yellow ones would be sister chromatids. These 2 green ones would be sister chromatids. But this green and yellow where they overlap, those are called non-sister chromatids. They come together, and what they do is they actually exchange some genetic information there. And so there's kind of this mixing up of the genetic information. Those homologous chromosomes, they line up in the center of the cell, and that's really the same process that we saw in mitosis, where they lined up in the center of the cell. The only difference here is that we have the homologous chromosomes lining up together instead of just a single duplicated chromosome set. All right, then we move on to anaphase. And in anaphase, these spindle fibers are connected to the chromatids, and they are going to start pulling them to either side of the cell. The difference here is that in anaphase, those sister chromatids, they remain attached at their centromeres, and they move towards either side of the poles. And so in mitosis, you saw that those sister chromatids separated, but here they stay together. So those have stayed together and moved to the side of the cell. Then we have the telophase, And so we see that this is a division right here, and we end up with 2 cells at this point. Okay? And those 2 cells have they have chromosome number of 46, but that's not really going to work for gametes, because remember, eventually these gametes might be used, the sperm might fertilize an egg, and if each of them had 46, then we'd end up with 92 chromosomes, and then it would keep growing from there. So this next part of this process, which is called the second meiotic division, this next part of the process is basically going to reduce that chromosome number to 23. That way all sperm cells have 23 chromosomes, all egg cells have 23 chromosomes, and when a sperm fertilizes an egg, that fertilized egg ends up with 46 chromosomes, which is how many people need to have. So this prophase 2 is actually very similar to what we saw in prophase of mitosis, because in this part the nuclear envelope is breaking down, the chromosomes are condensed, and in this part, in prophase 2 here, there is no crossing over like there was in prophase 1. So prophase 2 is actually more similar to prophase of mitosis. And then in metaphase, we have those chromosomes aligning on this, what we call a metaphase plate. They basically just line up in the center of the cell, like they did in mitosis. Okay, so in anaphase 2, what we see is those centromeres separate, and the 2 chromatids move to opposite sides of the cell. And so you can see here it's kind of small, but these chromatids have now separated, and they're each moving to either side of the cell. Okay. Then in telophase 2, we end up with the genetic material on either side of the cell. The nucleus is starting to reform. It's going to go through cytokinesis, and this is going to make 2 cells here. This cell is also going to make 2 cells, and so what we end up with is we end up with 4 daughter cells. And each of those daughter cells has different genetic material in it. So those chromosomes are different. Now, when we started out with a diploid cell. And I've been talking about the human chromosome set. So diploid is 46 for humans. That's how many chromosomes are in each cell of your body. In these gametes, they're haploid. And haploid means that there are 23 chromosomes in each of these cells. So our somatic cells are diploid, and our gametes are haploid. So the next thing I'm going to talk about is cytokinesis. And cytokinesis happens at the end of mitosis, it happens at the end of meiosis, and it's actually the division of the 2 cells that have formed. And this process is a little bit different in animal cells and plant cells, so it's worth mentioning how this occurs. So in an animal cell, after anaphase, these microfilaments form around the center of the cell. And microfilaments are basically what are going to pinch the cell in half. So they constrict, and they form what we call a cleavage furrow. It's like a little indentation in between the 2 cells. The microfilaments then completely pinch the cell into 2 different cells. In a plant cell, this is just a little bit different because these carbohydrate vesicles form around the center of the cell. Those vesicles, they fuse, and that actually starts to form the cell plate. So once that cell plate is completely formed, it separates the 2 cells, and then you have 2 different plant cells formed. And that's it for this lesson on cell division. I hope that was helpful.